So the recording has started. So again, welcome to this 12th and final video teleconference for BIOL 105YC. Um, this will be on the last topic before our exam, no? So the last coverage for our exam, which is on the immune system. Okay, so I will now begin my presentation. Okay, so if we look at our calendar, we are now on the 28th of April, and we are doing an extra video teleconference. Actually, this is the 12th one. And I looked at my calendar, I checked my calendar last night. I believe I told some of you that the final exam week is on the second week. Apparently, it's on the first week of May, no? But the registrar hasn't given any schedule yet as to when. So just stay posted with that. So our final exam for this will be multiple choice, and the coverage will be the topic after the semifinals. So it will not be comprehensive. So I think that will enable you to really study everything that's included in the exam. So let me. Okay, so are you still able to view my presentation? Visible pa ba sa inyong screen?
Yes, sir.
All right, sige.
Yes, sir.
Okay, thank you. So this morning we will be discussing, or it will be a lecture about the immune system, no, particularly the immune system of humans. Okay, so this is the last topic and the last coverage for our final exam. Okay, so when we speak of immune system and immunity, particularly in vertebrates like humans, there are two levels. Okay, so the first one is this innate immunity, okay, as we see here on top. And the innate immunity is composed of our skin, the barriers, okay, from pathogens, as you see here, no, those that can cause diseases like bacteria, fungi, and viruses. Then we also have mucous membranes now on the inside of our inside surfaces of our bodies, like inside your nostril, inside your nose, or inside your mouth. If you can feel those with your tongue, diba? The sides of your mouth, your cheeks, they are actually quite moist. And in your nose, there are mucus layers which actually help entrap bacteria and other pathogens, no? So that they will have lesser chances of entering your body. Then we also have these secretions. So a good example of this will be tears. Okay, so if you're not aware, our tears is actually equipped with antibodies that destroy pathogens like bacteria. So that's the reason why your eyes are able to be kept open without the risk of getting diseases right away, no, in your eyes. Because your tears, or the one that bathes your eye, the liquid, is antibacterial. Okay, so these are a part of the innate immunity, no, that we can see. Then we also have white blood cells, those that can eat, no, or phagocytose bacteria, fungi, and even viruses. Okay, and the inflammatory response that we observe every time we get an infection, no. So for example, naghubag imong tonsils, na kay tonsillitis, or nasamad ka dayon naghubag ang imong samad. Okay, those responses are actually part of the innate immune system. Okay, so what's noted here is you are born with this innate immune system. So maski baby pa lang, 1 day old, na innate immune system. Mao nang gitawag siya innate. And it's also quite fast in its response. So paspas ang iyang pag-response. So this is the default immune system that you have. So kung sa ato pa nagpalit kag laptop or computer, This is the default antivirus that the computer has. Okay, but even though it's fast, it's actually not as specific as this adaptive immune system that we have here at the bottom. Okay, so let's look at first the innate immune system. So what we see here is a diagram of hematopoiesis, no? The formation of blood cells. So not included in the immune system, of course, are the red blood cells that I'm encircling here. These are not included, and the platelets. But the rest are all white blood cells. So when we talk there about white blood cells, there are many, many types of them. Okay? The one here on top, which I'll encircle with blue, okay, these ones, eosinophil, neutrophil, basophil, mast cell, dendritic cell, macrophage, monocyte, they all come from this cell called myeloid progenitor cell. And they are all part of the innate, no, letter I, innate immune system. Okay, so take note of that. So these are the ones that phagocytose foreign bodies when they enter your body. No. However, important to note, there's one among them, the dendritic cell. So this one, this is not only part of the innate immune system, but it's also part of the adaptive immune system. It's a link. Okay, so let me just draw that there. So all of these encircled in blue are part of the innate immunity, okay, including the dendritic cell. But the dendritic cell may also function as part of the adaptive immunity response. So I labeled it here A. Okay, so later on we'll be looking at the ones at the bottom here. Okay, so let's start first with these, the ones on top, innate immune system. Okay, so far, are there any questions or concerns? Wala? Okay. So, if you've heard of leukemia, di ba, leukemia is also known as the cancer of blood, particularly white blood cells. Um, you see here two types of progenitor cells. So the one here on top is called myeloid progenitor cell. There is a type of leukemia called myeloblastoma. Okay? Meaning to say, the affected cells are the ones here on top, the ones that are functioning in the innate immunity. So, abnormal ang ilang growth because the myeloid progenitor cell is abnormal. So, that's actually a form of leukemia called myeloblastoma. Okay, so, just a side note. Okay, so how do these cells in the innate immune system detect foreign bodies? Okay, we have these intermembrane proteins, integral proteins known as TLRs. Okay, so what does TLR mean? It means toll-like receptor. Okay, toll, T-O-L-L. So, anong toll man? I imagine it's like entering a toll gate, 'di ba? So, not toll, but a TOLL gate wherein you need an ID or you need permission, a slip, before you're allowed to enter. So I think that's the reason why these proteins are named toll-like receptors, because they serve as a filter and as a detector for foreign bodies. So there are four of them shown here. First, we have TLR4, which is able to detect the presence of lipopolysaccharide, which is present only in bacteria. Okay? So when bacteria enter your body, they carry with them their lipopolysaccharide on their surface, and the TLR4 of your white blood cell, this one, will detect them, and your white blood cell will phagocytose, okay, the bacteria. Another toll-like receptor is TLR5 that detects flagellin. Okay? Flagellin is a molecule that's present in the flagella of bacteria or prokaryotic cells only. So, when TLR5 detects flagellin, that means there's bacteria present and your phagocytic cell will be able to eat the bacteria because it's detected by the TLR5. And then here we have other TLRs, TLR9, which is able to detect CpG DNA. Okay, these are DNA that have unusual ratio of cytosine and guanine, and they are unmethylated DNAs, and they are associated with fungi. So if fungi or pieces of fungus enter your body, so let's say in your nostril, okay, or in your lungs, this TLR9 will be able to detect the unique DNA present in the fungi and will allow this cell, this phagocytic cell, to actually eat the fungus, the fungi debris. Okay, so that's TLR9. And then lastly, we have TLR3, which can detect double-stranded RNA. Okay, now double-stranded is associated with DNA, not with RNA, but this one is double-stranded RNA. Therefore, it's foreign to our body. And in fact, it's only found in certain viruses. So when TLR3 detects double-stranded RNA, it can detect the presence of the virus and therefore will allow your phagocytic cell to phagocytose that particular virus. So these are all detectors present in your innate immune system, no, in your white blood cells. Um, Although they are specific to bacteria, to fungi, and to viruses, they are not that specific. Okay? So it's like if I'm gonna ask you, how do you distinguish an animal from a plant? Can anyone answer? Give me one distinction between an animal and a plant. Oh, that's an animal. Oh, that's a plant. Come on, based on cell, based on the cell of the plant and animal, what's the difference?
Sir, based on structure sa cells, sir.
Hmm.
They are different, po, sir.
Why?
And that,
Because
for plant cells, they are more sort of arranged like si tawag gani? Like blocks, sir. And then for animal cells, they're more let's say in the skin cells of animals, sir, they are like arranged in a flat manner, sir. Like
okay, so
on top of it.
that's on how they look like. But what's present in one cell that's absent in another? Plant versus animal.
In plant cells, sir, there is the presence of chloroplast, sir.
Okay, and cell wall, di ba?
Cell
So,
walls.
ah, so you're able to distinguish between plant and animal. So, mura kag innate immunity can distinguish between bacteria, between fungi, and viruses. However, if I'm gonna ask you between, if we say there's a cell wall, oh, it that must be a plant cell. How sure are you that if there's a cell wall that's a plant cell, or what type of plant is that? What species? Diba? So for example, fern versus pine tree, they're both plants. They both have cell walls. How do you distinguish a fern from a pine tree by just looking at the cell? So you see, ang innate immunity, they're able to distinguish plant from animal, for example, but they cannot distinguish the more minute differences between different species. Okay? In other words, what I'm trying to say is the innate immune system can be specific, but it cannot be that specific. Okay, so that's the point. So the specificity is thanks to these toll-like receptors. Okay? But they are very limited. Okay, as you can see here. So That's one disadvantage of innate immunity. Okay, not very specific. Um, both for the innate and the adaptive immunity, now the cells, the white blood cells, that's what I'm talking about here, are circulating around your body not just through your blood vessels, but also through the lymphatic vessels. No? So this is your lymphatic system. So aside pala from your veins, arteries, and capillaries, there are networks of tubes, no, or systems in your or vessels in your body that exclusively carry lymph. So lymph is like the plasma portion of your blood where they contain the white blood cells. Okay, so as you can see here, they are intertwined with the blood vessels. No, kaning green lymph vessel, while the red one are blood vessels. So they are dispersed throughout your body, and then there are certain headquarters for white blood cells, no? Like your tonsils. Okay? Your tonsils are actually headquarters for white blood cells. So they stay there. And if there's an infection, the tonsils will be swollen because they are working double time. The headquarter is working double time to kill certain pathogens. And then we also have important organs like the thymus, no, found here on the neck or in the neck. We have the spleen and even your appendix functions, no, in the lymphatic system, part of the immune system. Okay, so this is where the white blood cells roam around. No, they patrol your body via the lymphatic vessels. So here's another look at the innate immune system. No, so for example, nasamad ka, okay, and then you did not clean the wound, so bacteria enter the wound. There are actually waiting white blood cells called macrophages under your skin that will react and eat the bacteria. And at the same time, they will also release chemicals to signal other white blood cells about the infected area. And then we also have this cell called the mast cell, which releases chemicals in the infected area, which causes the blood vessel in that infected area to dilate. Okay, so the diameter of the blood vessel will increase to allow more blood to flow and to allow white blood cells to pass through the barrier of the blood vessel, no, into the infected area so that they can um, do their work and kill the bacteria that have accumulated in the infected area. So this is one reason why if you have an infection, no, or na naghubag sa imong body, it appears reddish. That's because the blood vessels in the area are actually dilated thanks to the signal from the mast cell, which releases histamines. Okay? The same chemical that is also triggering allergies, di ba? Mao nang mamula pug kung ma-allergy ka. Okay, so if you try to feel infected areas or kanang naghubag nga area, they also feel warmer than the rest of the body because there's also more blood flowing through them than the rest of the body. Okay, so this is actually very fast. No, in a matter of seconds, white blood cells can already act upon in the area. So that's an advantage of the innate immunity. Okay, so here we can see the white blood cells are finishing up the bacteria and they die after they do the killing, no, and the bacteria die with them. So we usually see this no, in a form of pus, no, kanang nana on the infection site, no, kanang naghubag na site. And if you leave it alone by itself, it can be reabsorbed back by your body. Okay, kaning mga dead debris of white blood cells and bacteria. So this is the innate immune system working, no? So mga infection site, and then there is kanang naghubag sa kanang English ana kalimutan ko. Sukat the term inflammatory responses. I'm sorry. So kana naa kay inflamed area, so naghubag imong ngipon, nagsakit imong ngipon. So what's working there is actually your innate immunity. Okay? So that's innate immunity for you. Now, innate immunity is not enough. No, as I said, we are born with this. So even babies, pwede sila magka have inflammatory responses. No, ang mga babies. So, but it's not enough because it's not that specific, and other types of pathogens can still get through the innate immunity. So there has to be a second level only for vertebrates, no, including humans, called adaptive immunity. So this one is much slower in response, and it takes time to form in the body. So that's why babies are not formed with this. But if you let babies play and they explore their environment, their adaptive immunity can actually form slowly, no? This is also one reason why we allow babies to explore, and you don't just um, put the baby in an environment that's very clean, because otherwise the adaptive immunity of the baby will not develop in such clean environments. No, dapat maghugaw-hugaw so na siya ginagmay. Okay. So there are two strategies of this adaptive immunity. It can be a humoral response, wherein antibodies are involved. So the term humor means body fluid, diba? So body fluids like your plasma or lymph, that's humoral response. And then there's also cell-mediated response, wherein the cells involved are known as the cytotoxic T cells, okay? And they kill cells that are actually infected by the pathogen. Okay, so back to this illustration. So now let's focus here on the one at the bottom. Okay, so all of these white blood cells, which originate from the lymphoid progenitor cell, are part of the adaptive immune system. But there is again an exception. Okay, and what's the exception? The exception is the natural killer cell. The natural killer cell is an exception because it's part of the innate immune system, and it works in killing tumor and cancer cells, kaning natural killer cells. So ultimately, what's really part of the adaptive immunity are just the T cells and the B cells, which can become plasma cells. Okay, so please remember this one. And because their origin is the lymphoid progenitor cell, they are also known as the lymphocytes. No, lymphocytes. Lymphocyte. So, nakay T lymphocyte and you have B lymphocytes. Okay? And there's also another form of leukemia that can affect this lymphoid progenitor cell. So, we call it lymphoblastoma. And people with lymphoblastoma have okay innate immune cells, but their adaptive immune cells are abnormal. So that's lymphoblastoma as opposed to myeloblastoma. Okay? So as you notice here, there's always exception to the rule, no? In the innate immune system, the exception is the dendritic cell. In the adaptive immune system, the exception is the natural killer cell. So thing to remember. Okay, so here's a closer look at the B and T lymphocytes. So what is drawn here are the receptors, no, on their cell surfaces. So these are made of proteins, and there is a difference in terms of shape. No, in the B lymphocyte, the receptor is this Y-shaped thing. Okay, while in the T lymphocyte, the receptor is linear, so straight lang. Okay, and they are found on the surfaces of these B and T lymphocytes. So another thing to note is the name. Why is this called B and this one T lymphocyte? And the reason is both of these are made in the bone marrows, but only the B lymphocyte matures in the bone marrow. So it's called a B cell after bone marrow. While the T lymphocyte needs to migrate first to the thymus, di ba, katong naa diri sa neck, and it matures there. So that's why it's called the T lymphocyte or the T cell after thymus. Okay, so these are the two players of the adaptive immunity, B and T lymphocytes. Okay, closer look at the receptor of the B lymphocyte. So this is the Y-shaped thing. There is a C region, okay, a constant region. Okay, before that one, I'm scribbling a lot, so let me erase that. Before that, there's something to note. No, the one here in the middle, the long ones are known as the heavy chains. And here on the outside, the shorter ones are known as the light chains, okay, of the receptor, okay, antigen binding site. What's more is you also have these C or constant regions. And the variable regions are the ones here at the tip. So kanina sa mga tumoy nga regions which help detect the viruses, bacteria, and fungi are variable, meaning to say maglahi-lahi na sila. Okay, they can vary from one type of B lymphocyte to another. So this is how it works, no? When the B lymphocyte detects the presence of a pathogen, no, by binding to the antigen, specifically the epitope, kaning the tip portion of the antigen, when the antigen receptor binds to that, the B lymphocyte will actually be able to detect it, and it will differentiate into a cell that is able to secrete antibodies. So kaning antibody is just like the antigen receptor, but it's soluble, meaning to say it can be thrown away, murag boomerang. No, that's how I imagine this. Here, the B cell is like holding on to the boomerang. But once the B cell detects the pathogen, the B cell will have the ability to throw away the boomerangs, okay, to target the pathogen. And the boomerang is known as the antibody. Okay, so the antibody can stick to the antigen and disable or even kill the antigen. Okay, and here we have the detail for the antigen binding site of the T cell. So it's more linear, but the same story, the tips are all variable regions, no, as you can see here, while the base is the constant region. So kaning base is constant. So what the T cell does with that is, here in this illustration, we have a cell that is infected. No, kaning purple, this is a host cell. It's infected by this pathogen here. The pathogen is able to enter the cell. So this is quite too late for antibodies to work on the antigen because it has already entered the cell. So what the host cell does is it uses a molecule called a major histocompatibility molecule to present, to display the antigen inside of it. Okay? In other words, the host cell here is like someone asking for help, okay? By showing the evidence that they are infected, no, using the major histocompatibility molecule, and it will be the T cell that will be able to detect that. Okay, the T cell will detect that, and once the T cell detects that, the T cell will cause the death. So iyan yung patyon ang cell na na-infected. Okay, the T cell will kill the infected cell. So that's why we call this as the cell-mediated response. Okay, so this is just a detail of the major histocompatibility molecule. Okay, so any questions so far? For those who are new with this topic, I'm sure you are sort of confused already. Okay, so if there are questions, please just don't hesitate to stop me so that you can ask your questions. Are there any questions so far? Wala? Okay, so let's move forward. Then later on, we'll review. Okay, so since we are talking about adaptive immunity here, di ba? Adaptive man. So, meaning to say, the B and the T cells must be of different types in order for them to be able to detect different types of pathogens, no? So that means their diversity has to be really, really great. And how is this generation of diversity of B and T lymphocytes done? Okay, the answer is in the gene that makes the antigen receptor that we see here at the bottom. Okay, as shown here, the gene that makes this receptor consists of variable exons, di ba? Exon ang tawag ani, expressed sequences. Look at them from V1 to V40. 40 different variable regions. What's more is there's also a portion of the gene that makes the junction region. The junction is the boundary between the V and the C. As we see here, kaning last nga. And then you only have one copy of the C or the constant region. So this is the gene that is used as an instruction to make your antigen receptor. Okay, so with the help of an enzyme called a recombinase, mura siya lottery, murag loto, no? Out of V1 to V40, only one of these will be chosen. Okay, so in this case, si V39 ang iyang gipili. No? So random choosing. And out of the J1 to J5, only J5 is chosen. And of course, the constant is always a constant. So you can already imagine here, no, in this particular stage, how many combinations can be made. So pwede V1, J2, and then C. Pwede V20, pwede J3, and then C, and so on. So literally, you can come up with thousands of combinations. Now, if you use mathematics or statistics to compute, so moabot gyud siya thousands, no, or more combinations for this. Okay, so once this has been recombined, an mRNA will be made, no, it transcribed. So you have here the V39 and J5 transcribed. And the messenger RNA will be translated into the protein that we see here that has the V. The V is made from the V39 and J instruction, and the C is made from the C. Okay? And that's what you have here in this light chain. No? Okay. So, can you imagine pila ka combination, no? Maglahi-lahi ang combination ani plus na pag siya other combination nga variable. So you can really have perhaps millions of combinations of these. So that's how the diversity in the B and the T cells are generated by random generation of the genetic code that instructs for the making of the antigen receptor. Okay, so because it's made randomly, katong mga B and T cells, it's possible that the body will be able to make a B or a T cell that is reactive against itself. No? So the body has to be able to choose which T or B cells will be allowed to live. So here's an example. Okay. So in this first scenario, the one here on the left, your body will test the resulting T or B cell against itself. So in this case, against the thymic cell, no, because this is a T cell. And in this case, the fit between the receptor of the T cell and the antigen on your thymic cell is too good, so that it's a strong affinity and your body will choose that T cell to die. Dili siya ipabuhi sa imong body, because otherwise, if this survives, this will cause excessive autoimmune diseases. No, kay ma-recognize man niya imong own cell too well enough. Okay, and then here in the middle, the body tests the T cell against itself, and there is no reaction at all, no affinity. So the body will also choose this cell to die. Useless man siya, kaya di man siya makadetect og antigen. And here on the right, we have a T cell that has a weak affinity with the antigen on the thymic cell, and so the body will choose this to survive. So it's like recruiting for an army. and then you have an audition for the army. Those that are too good, you don't accept them because they can be a threat to your government. Okay? Basig na kudita because of the too good army. And then here in the middle, if you recruit an army, then some of them are no good at all with weapons. They don't know what to do at all. Of course, you don't want to accept them. They're useless in the army, that is. And then here, the last one, you recruit an army and okay, they know how to handle themselves, but they are not that good. They can still be trained. That's what you want. And that's the same strategy that the body is using to choose which T or B cell will survive. Diba? Okay, so I have a question for you. If you can remember, why does our body need to choose which T or B cell will survive? Okay, give me a short answer. Why? Or any guess again? Nganong kinahanglan mapilion pa sa imong body kinsay mo survive? Nganong dili na lang tanan?
Cancer because uh, the T cells that are example like too good at recognizing other cells may cause autoimmune diseases, sir. And also the T cells that are not good at all um, essentially cannot do anything good, sir.
Okay, that's correct, no? Okay, so I think you can imagine now for those people who have autoimmune diseases, this could be where the defect is, no? Kaning pagpili sa ilang mga T and B cell is not properly done so that those B and T cells that are too good are allowed to live. So mao nang napod sila autoimmune diseases, they're prone to that. But I have another question from that. Why is the body making the too good and the no good B and T cells? Nganong dili na lang man nga kaning middle lang iyang himuon? Nganong apil man ning duha? I mean, does our body have a choice of just making the weak affinity na T or B cell? Yes or no? Anyone? Na by choice? Oh, para ato tong ganina nga nag-recruit ta sa army, na ba tay choice kung kinsay mo abot sa atong recruitment katong maayo, katong dili maayo, or katong wala gyoy hanaw? Do we have a choice in that?
Wala, sir.
Wala, di ba? And the reason for this no in our immune system is we go back to this story, kani ba? The gene that is used to make the antigen receptor on the B and the T cells is composed of these variable segments. Tapos, by chance, murag loto, murag lottery, isa lang ang pilion from V1 to V40. So, and J pagyod from J1 to J5, isa ra pod ang pilion. So, it's probable that during the making of this antigen receptor, ang antigen receptor nga resulta is too good or no good at all. So mao na ang reason why your body needs to undergo kaning selection, no? Clonal selection of T or B cells. Okay, so that is an important thing to remember about adaptive immunity. Okay, so now nga nakapili na ta og B og T cells nga mo survive, the next problem is how do we increase their number? Okay? So, nagpili ka kinsay mabilin, next problem is unsaon nimo sila pagpadaghan, proliferation. Okay. So, here's an illustration, no? So, you have three choices, for example, out of millions. Then you have here na ay virus or bacteria nakasulod kaning antigen. So, what your body does is it will try to fit. So murag si Cinderella ba nga naay sapatos na bilin, kinsa nga babayi nga maiguan ani nga sapatos. Okay? So in this case, antigen, kinsay antigen receptor ang maigo ani. Siya ang pilion. Diba? Kaning nasa tunga. The other ones on the left and right are not effective against this antigen. So only the one in the middle is selected, clonal selection, so that it can be proliferated. Okay? And what's more is half of them will serve as a reserve. No, reserva sa army. We call these as memory cells. And half of them will be the fighters. Okay? Ang musulong katunga lang, ang katunga reserba. Okay? The reason for this memory cell is because the next time your body will encounter this antigen again or this pathogen again, you still have a reserve in you para mas paspas ang pag-react sa imong adaptive immune system, no? So that's also the reason why vaccination is done, so that your body will create memory B or T cells. Okay, so mao ni clonal selection. So pilion kung kinsa ang mag-fit best sa antigen, siya day ipadaghan. Half of them will be reserved, half of them musabak sa giyera. Okay, so that's the story here. And here we have a graph showing the adaptive immunity no, on the first infection. Look at the increase of antibody A here. Okay, ni-increase siya pero hinay-hinay, then dili kay daghan. No? But after the first infection, remember there's already a memory cells. There are memory cells, B and T cells. So the second time around, ma-infected ka, say with a flu virus, mas paspas ang response sa imong adaptive immunity in the making of antibodies and mas daghan po diyang mahimo. No? And here we have new infection of a new type of disease. So magsugod na po og hinay ang response sa imong adaptive immunity. And on the second attack, mas paspas na po diyang response. No? So that's how our adaptive immunity works. Okay. So before that, we only talked about the B and the T cell. No? There is one cell under T. T gihapon siya kay direct man ang iyang pag-detect sa antigen, no? And it also matures in the thymus. We call this the helper T cell. Okay. Kaning helper T cell, no, will be able to detect the presence of pathogens by detecting the antigen that is presented to it by this cell. This is a dendritic cell, part of your innate immunity. You know, look at the shape, it's dendritic cell. So the dendritic cell will tell the helper T cell that there is a pathogen. Okay, what's gonna happen is a chemical called cytokine is going to be secreted by both the siya tawagan na nga cell dendritic cell, okay, and the helper T cell. So they will release cytokines which will stimulate the helper T cell itself to proliferate. Mudaghan ang helper T cell. Okay? And when the helper T cell increases in number, it will help. Mao na ang tawag sa helper. Helper only but not killer. Okay? It will help or empower the B cell and another type of T cell called the cytotoxic T cell, no? And this is where our humoral immunity works, no? Kaning B cell tungod sa antibodies and cell-mediated immunity by the cytotoxic T cell. So you have the helper T cells first. And in people with HIV, the target of the virus is the helper T cell. So kung na HIV, ang helper T cell, mga matay, no? Kay sila man ang host cell sa virus. And therefore, ang affected nga immune system with people with HIV is the adaptive immunity. Maunang, they are very prone to new infections, no? Because their adaptive immunity is compromised. So maunang nana dayoy acquired immunodeficiency syndrome, no, acquired immunodeficiency syndrome or AIDS if you have HIV. So this is just a sequence showing how the helper T cell here, okay, detects the presence of the pathogen and then releases cytokines to stimulate the B cell to proliferate. Half will become memory B cells, while half will become the plasma cell, the army, that will be able to release antibodies, okay, which will neutralize the antigens and the pathogens. So these are the three ways by which the antibodies work. It can be neutralization, no? They will surround the virus in this example, thus preventing the virus from infecting other cells because it's being surrounded. Number two, they will also prevent the movement of the pathogen. So the pathogen will just stay put, and a white blood cell called a macrophage will go to the area and eat the pathogen. So that's another function of the antibody. And the third function is this. The antibody will serve as a base for a turret. So meron siyang turret gun, no? Okay, assemble. We call this as the complement protein, no? Ang base niya kay antibody. And the complement protein will form a pore on the membrane of the bacterium or the fungus so that the bacterium or fungus will lyse, will die, or will burst. Okay, so these are the three main modes of action of the antibody released by the plasma cell, which was originally a B cell. Okay, so remember that one. There are many types of antibodies, no? The ones that's attached to the B cell is called the immunoglobulin D. So mo na ang another name for antibody, immunoglobulin. The ones that are released by plasma cells are either IgM or IgG. And then IgA is present in the tears. Um, IgE is going to bind to mast cell to stimulate it to release histamine. So usually, kanira yung IgM o IgG ang ginagamit to detect the presence of pathogens. Di ba? Katong rapid test bitaw sa COVID-19, di ba? It's IgM and IgG ang ginadetect, the release of these antibodies. Okay, so here's a graph showing that in the release of IgM or IgG, it's IgM first that will be released. No, si IgM first, and then later on, IgG will follow. So in the rapid test, these are the ones that are detected. And here's the mode of action of the cytotoxic T cell. No? So the cytotoxic T cell is empowered by the helper T cell. It's going to go to the cell that's infected. No? This is the host cell. So na siya mga antigen sa sulod. So it's going to detect the antigen with the help of this major histocompatibility molecule from the infected cell. And once it does that, it will release enzymes and perforins, which will cause the host cell to burst. Okay, so this host cell will die and the antigens will be released so that the antibodies from the B cell will be free to bind to the antigen. So that's how the cytotoxic T cell works. So here's a summary. So you have here the antigen. If the antigen is present, okay, if the antigen is present, it will be eaten by antigen presenting cells, which will be detected by the helper T cell, which will empower either B cells or cytotoxic T cells, as you can see here. Okay, and then the B cells are going to proliferate. Half of them will become memory B cells, while half of them will become plasma cells, which can secrete antibodies. Okay, while for the cytotoxic T cell, half of them will become cytotoxic T cells to kill the host cells that are infected, and half of them will also become memory cytotoxic T cells. So that if there's a second infection, No, it will be the memory B and the memory T cells that will act upon on the second infection to release new plasma and cytotoxic T cells. Okay, so this is the mode of action of the immune system, the adaptive immune system. So this is when you have the disease, but since we don't want to have the disease first in order for us to be immune, the answer is vaccination. So vaccination. So in vaccination, you are given an attenuated virus or pathogen to stimulate your body to create memory B and memory T cells so that when the real pathogen infects you, you will have a faster adaptive immunity. So mao na ang role sa vaccination. You don't have to wait na masakit ka before you become immune. So have vaccination. So mao na yung vaccination no, for the SARS-CoV-2, the one that we hear right now. There's antigen vaccination. Okay, where our proteins or attenuated viruses are injected to the patient. We also have RNA or DNA vaccines, wherein the RNA is injected so that it can produce the proteins of the virus in the cell, or an antibody from someone who has been infected and survived can be injected to the person. Okay? But there is a disadvantage. Pag antibody lang ang i-inject, no? Meaning to say, diretso diri sa ubos ang i-inject sa imo, your body will not be able to produce the memory B and memory T cells. So, short term lang ang imong immunity kung ang antibody lang ang i-inject. So, that's the disadvantage of this. So, just a reminder, if you get vaccinated, it's not a guarantee that you will not get sick. But instead, it's a guarantee that whenever you get the sickness, it will not be as worse as someone who has not been vaccinated. Okay. And then I'll leave this to you to read, no, kaning allergies, okay, versus autoimmune disease. So an example of the autoimmune disease is the SLE in your laboratory activity, katong systemic lupus erythematosus, which is an autoimmune disease. And a while ago, we talked about the implications ng anong na autoimmune disease, katong ang pagsala sa imong T ug B cells wala natarong sa imong body. Okay. And this one ends my lecture on the immune system. So I think it's quite comprehensive, but it's not everything. So the goal is, if you read your book, you will be able to understand better, no? Because at least you have heard the terms once already. Okay. So are there any questions or concerns? Time check, 9 a.m. In 10 minutes, I think all of us or most of us will meet for genetics again. So I hope you understood lang guys, because this topic on the immune system, I think is a very challenging one, no? Even to biology students. Marag, isa ni siya sa pinaka-challenging topics in biology. Lisod sabto ng immune system, how it works. But once you get over it, I'm sure you will have a hard time forgetting about it also. Okay, so, no questions? All right, so you just need to wait for your final exam, no? I-announce pa man sa registrar kung kano sa atong schedule. And if ever it's not announced, I think we can still have up to the second week of May for our exams, no? So the coverage will be the one after the semifinals up to the immune system. All multiple choice with one choice each. Okay. So if there are no questions, guys, I think we have to end now to give ourselves enough time to prepare for the next video teleconference. So thank you very much for joining in this last video teleconference for BIOL 105. And I hope you learned something in this semester. And see you later in the next video teleconference for genetics. And you are now all dismissed.
Thank you, kayo, sir.
All
Thank you,
right, you're welcome.
sir.
Sir, thank you kayo.
You're welcome.